Okay, now we're going to look at the chemical synapse, which is the majority of the synapses in the nervous system. And we need to remember that they are different from electrical because they don't lose any signal strength as they move. But what happens there is it's a lot more complicated neuromuscular junction, because you can have multiple neurons that are secreting different types of neurotransmitters, some of them excitatory, some of them inhibitory. But these are the steps. First of all, the action potential gets down to the presynaptic neuron, and it triggers the voltage-gated calcium ion channels down in the axon terminal to open up. And all the calcium ions flood in, and that causes the synaptic vesicles to release the neurotransmitter into the synaptic cleft, and they do this by exocytosis. Then the neurotransmitters bind to the receptors on the postsynaptic neuron, and this opens ion channels, and that leads to a local potential, and if it reaches threshold, it'll go ahead and become an action potential. Now, postsynaptic potentials are local potentials that occur in the membranes of the postsynaptic neuron. And as the membrane potential in that postsynaptic neuron gets closer to threshold, it causes a small local depolarization. And that is because either the sodium channel's open or the calcium channels are open. And it's called an excitatory postsynaptic potential. See, over here, it's moving closer to threshold. Here's threshold, and it's getting closer. It's moving closer to threshold. And that's the excitatory postsynaptic potential. The membrane potential moves farther away from the threshold, and that can cause this depolarization here that you see. That can cause that, and that's either because the potassium channels or the chloride ion channels are open, and that's called an inhibitory postsynaptic potential. Now, synaptic transmission can be terminated just by ending the effects of the neurotransmitters, and this can be done three ways. Sometimes the neurotransmitters just diffuse away from the synaptic cleft into the extracellular fluid, and then they're reabsorbed into the neuron or an astrocyte. Another way is it can be broken down by enzymes, and the byproducts of that reaction are then reabsorbed by the presynaptic membrane for assembly of that original neurotransmitter that you started with. The third way is some of the neurotransmitters are reabsorbed into the presynaptic neuron by a process that's just called reuptake. So the transmission is going to continue until we get rid of the neurotransmitters. And these are the three ways. It'll diffuse away, it will be broken down by enzymes, or it's reabsorbed by reuptake. Now, almost all neurotransmitters have a similar pattern of use, and there are 100 different known neurotransmitters. They have similar features. They're all made in the cell body or the axon terminal, and then they're put into the synaptic vesicles. They get released from the axon terminals in the presynaptic neuron. They cross the synaptic cleft, and then they bind to specific receptors on the postsynaptic membrane. And the effects are rapidly terminated by removing or degrading the neurotransmitter. But no matter what type of receptor, the binding of the neurotransmitter to the receptor leads to either the excitatory effects, EPSP, or the inhibitory effects, IPSP. Most of the neurotransmitters have both effects, and it just depends on which postsynaptic receptors they bind to. And they can have one neurotransmitter can have several different types of receptors. But we classify neurotransmitters into 4 groups based on their chemical structure. First is acetylcholine, and this is the one that we talked about that was used to stimulate the skeletal muscle fibers. It's widely used by the nervous system. There are cholinergic synapses that use this acetylcholine, and we find them in that neuromuscular junction that we studied in the last unit, and we also find them within the brain and spinal cord and within the autonomic nervous system. They're mainly excitatory, but it can have some inhibitory effects, and it's quickly degraded by that acetylcholinesterase, that enzyme in the synaptic cleft. And then the byproducts of that degrading process are then recycled back into the presynaptic neuron to be used again. The second group are the biogenic amines, and these are a class of 5 neurotransmitters that are made from amino acids, and they're used extensively throughout the central nervous system and the peripheral nervous systems. They regulate homeostasis and cognition. The first 3 form a catecholamine subgroup, and they're all made from the amino acid tyrosine, and they're mainly excitatory. Within this group, we have norepinephrine, and we find this mainly in the autonomic nervous system. It influences your heart rate, your blood pressure, and digestion. And then we find it in the central nervous system, regulating your sleep-wake cycle, your attention, and your feeding behaviors. The second one in this group is epinephrine. We also call it adrenaline. And this is again used in the autonomic nervous system. It functions similarly with norepinephrine. This is also a hormone that is widely used in the endocrine system. The third group is dopamine, or the third one in this group is dopamine. And this is used extensively in your central nervous system to coordinate movement. It's involved with your emotion and motivation. And then the 2 other ones that are in these biogenic amines that are not catecholamines, serotonin, it's made for the amino acid tryptophan. And we find most of the neurons that secrete serotonin in your brain stem. And then the axons project all over the brain. It helps with your mood regulation, with your emotions, your attention, your daily rhythms, and your feeding behaviors. And then the last one is histamine, that is made from the amino acid histidine. And it is involved with arousal and attention. And outside the nervous system, it's a mediator of allergic responses. Then you've got your 3 main amino acid transmitters. Glutamate, this is the most important excitatory neurotransmitter in the central nervous system. And it generates the EPSPs, the excitatory, in the postsynaptic neuron. Then you have glycine and GABA that are inhibitory, and they induce the inhibitory effects on the postsynaptic neurons. The last group of neurotransmitters are the neuropeptides. And this is a group of neurotransmitters that have a wide variety of functions. They're made in the cell body and then carried to the axon. There's substance P that's involved with pain and temperature, your opioids like your endorphins that will cause pain relief. You oftentimes hear about somebody that's an avid runner, that they run so long until they have this endorphin release. This is a high that they experience. And then there's neuropeptide Y that's involved in your feeding behaviors. It helps you to feel either hungry or full.